Welcome to our virtual tour of alzdata.io, a new resource that's contributing to our understanding of Alzheimer's disease, AD, at the cellular level. Let's start with the homepage. It provides comprehensive background on the remarkable collection of legacy biopsy brain samples from AD patients. These samples, taken by leading AD researchers in the 1960s through the 1980s, are truly unique. They've been preserved with near-perfect ultrastructure, allowing us to see detailed cellular features that were previously unavailable to the scientific community. Moving to our Discovery page, you'll find information about the cellular features and discoveries that motivated the creation of this database. We've focused on early-onset AD cases, where we can observe cells affected by AD to different degrees right next to healthy cells. This provides novel insight into the disease's progression. You'll see details about paired helical filaments PHF, now known to be largely composed of tau proteins, as well as amyloid accumulations and modifications to subcellular organelles and cytoskeletons. Now, let's dive into one of our key features, the Analysis page. This section connects you to various web pages showcasing results and datasets derived from our image data. We've employed cutting-edge techniques, including serial block-phase scanning, electron microscopy, and deep learning-based approaches, to extract detailed information about interorganelle associations and cytoskeletal reorganization linked to neuronal degeneration in Alzheimer's disease. One of the powerful auto-segmentation tools we closely collaborate with is CDeep3M, an open-source deep learning tool for large-scale image segmentation tasks. By clicking the CDeep3M link, you can explore its suite of deep learning utility tools. On the Collected Data page, you'll find a hierarchy of datasets presented in a tree structure. The first level of the branches represents the neuropathologist's name. Our large-scale 3D data dissemination is based on an exceptional collection of legacy human brain biopsy samples from Alzheimer's disease patients. These samples were meticulously collected and preserved for EM analysis by renowned neuropathologists Drs. Robert Terry and Suzanne Mira in the 1960s and 1980s, respectively. The second level of the hierarchy represents the biopsy block sample label. A biopsy block sample is a tissue specimen obtained through a biopsy, a medical procedure in which a small tissue sample is removed for examination. After extraction, the tissue is processed into a block to facilitate detailed analysis. The final level of the tree presents the image data, which are collected for various types of analysis. A 2D survey image provides an initial overview of the specimen, enabling researchers to assess its overall condition and identify areas of interest for further study. A region of interest (ROI) within a 3D volume refers to a specific subset of data that researchers focus on for detailed investigation. Some of our ROI 3D volumes highlight key Alzheimer's disease features such as the PHF array and lipofuscin. In the Derived Dataset section, you'll find the results generated by our deep learning tools, including detailed 3D reconstructions and quantitative analyses of cellular structures. These tools, powered by deep learning-based computer vision, have been applied to segment and analyze key cellular components such as mitochondria, endoplasmic reticulum, synapses, and Alzheimer's disease-related pathological structures, including paired helical filaments, amyloid beta clusters, and dystrophic axons. Clicking the 3D object icon will launch our 3D rendering tool for interactive visualization. You are currently viewing the 3D rendering of an astrocyte cell process. The green mesh represents the astrocyte's plasma membrane boundary, while the yellow spheres indicate subcellular glycogen deposits. The blue structure corresponds to paired helical filament, PHF, array number 1, and the orange represents PHF array number 2. This 3D rendering tool allows easy zooming in and out using the mouse wheel, and you can rotate the object by holding the left mouse button and dragging left or right. Additionally, you can toggle the display of cell components in the 3D viewer using the checkboxes on the left side. On the image page, each image we produce is meticulously stored with comprehensive metadata, which is essential for comparing results across different experiments, sample preparations, imaging methods, and biopsy sources. A digital object identifier (DOI) is generated for each image. More importantly, all of our data is securely safeguarded by the UCSD Digital Access Management System and preserved in the long-term digital archive, Chronopolis. By hovering over the Image Data Download Options icon, two drop-down options will appear, allowing you to download either the display image or the full-resolution version. Additionally, the license information is displayed below the download options. Clicking the Detailed Viewer button launches our customized image viewer, which enables interactive exploration of complex 3D datasets. You can seamlessly navigate through cellular layers, zoom in on specific organelles, and compare healthy cells with those affected by Alzheimer's disease. Additionally, you can use the annotation toolbox on the left to mark your region of interest, ROI, and add metadata. This annotated ROI can be easily discovered using the search function, and its precise location can be shared via a URL. For more information, please visit our About Us section. You can also visit CIL.org for additional examples of biological image data. We hope you find alzdata.io to be a valuable resource in the fight against Alzheimer's disease. Thank you for joining us on this tour of a transformative tool in neuroscience research.